Hi, welcome to our Pharma Topics channel. Let us see about gene, which is very important. So it is the basic and functional unit of heredity. Actually, Graham Mendel first described about the gene in the year 1860, but he never used the term gene. He just used the term factor. Then Wilhelm Johansen coined the term gene in the year 1909, and he said it is the determinant of inheritance. The number of genes are always greater than the number of chromosomes, because all the chromosomes they carry number of genes. Genes are arranged in single linear order like beads on a string. So genes occupy a specific position in the chromosomes and they are called locus. So genes may exist in several alternate forms which are called alleles. So one gene codes for one polypeptide protein. Uh, if the protein is having a 3 or 4 chains, there will be 3 or 4 genes which are responsible for that. So DNA is the genetic material. Then what is a genetic code? Argobin Corana and Francis Crick and even many other scientists have contributed to decipher the genetic code. Actually, it's a triplet code. It is the relationship between the nucleotide in the genes in the the mRNA and the amino acids in the proteins, they encode. So there are 64 possible triplets. 61 are used to represent the amino acids. There are three digits. For example, three triplet codons are for termination. Since there are only 20 amino acids, uh, most of them are encoded by more than one triplets. There may be many triplets, but actually one tRNA codes for only one amino acid. The code is always in fixed direction from 5' to 3' direction. AUG has a dual action. It acts as an initiator codon and also amino acid methionine. UAA, UAG and UGA are stop codons. Among them, UAG can also code for tyrosine and UGA can code for tryptophan. And when they terminate the polypeptide chain, it is a nonsense codon. Let us know about the Wobble hypothesis. This hypothesis was proposed by Crick in the year 1966. According to this hypothesis, uh, first two base pairs are complementary, perfect complementary, but the third base may not be like that. The third base is called a wavering position or a wobble base. The actual base pairing occurs, as I said, for the first two positions only. So let us look at this example. G pairs with C. From the DNA, the GAG code is available. From this, mRNA is formed. A pairs with C. That is perfect pairing, and here perfect pairing is G with C. But the Wobble hypothesis says the first two pairs may pair complementary perfect pairing, but G may pair with any other which is non-complementary also. That pairing is called Wobble hypothesis. That is the reason there are 61 triplets. So let us see the genetic code library, the triplet code library. It will, you can see this table, it will appear like this. So all are triplets. Let us see an example. Let us say this is a mRNA, which is AUG actually codes for methionine, that's the start codon, and ACG codes for threonine, GAG codes for glutamate, CUU codes for leucine, CGG codes for arginine. You can look at the table also, and AGC codes for serine, and UAG is the stop codon. So this is how the deciphering of the code to the corresponding amino acids, and this happens in the ribosomes. So it's a short nutshell information of the gene and the genetic code. I hope it will be useful for you. Thank you for listening. Happy learning. If you like this video, press the like button. Kindly subscribe to our Pharma Topics channel. Thank you.